very much metastatic. So whenever it metastasizes from one place to another one, it's very difficult for the clinicians to target those cells to find out whether they will metastasize or not. So these are the points. But still we are working on something that if the cells metastasizes, then what happens? It goes to some other places. It is very much vigorous, and they have different properties like migration, innervation, circulation through the blood vessels, extravasation, colonization, proliferation, and angiogenesis, and then formation of a new tumor on other places at different parts of our body. So when it metastasizes, generally the present modalities that are, if it is present in a specific place, then surgery. If it goes through some other places, then chemotherapy, radiation, hormonal therapy, all these are present. But all these things, generally chemotherapy has a very bad side effect. So what is that? So whenever chemotherapy is done to some patients, doxorubicin, paclitaxel, and methotrexate, these are the main drugs that are being used nowadays. Along with that, there are a lot of side effects of these chemotherapeutic drugs. So to reduce the side effects, we need a lot of vitamins, a lot of this and that, and good food and those things. So if we can target cancer cells by some molecules, so these are the side effects. And if we can target those cancer cells with some molecules which are non-toxic to our normal cells, then it will be very good for us. So we are very much confused to which way to look. So towards the activity of the drug or towards the safety of the human being. So doing these things, I found that the dietary plant products that are present in our India, specifically all over the world they are present, but in India they are being used very much in food. So there are some benefits of using those things, but you may ask some question that, so why the prevalence of breast cancer is so much high in India? But there are some problems with these phytochemicals. These phytochemicals, whenever they are present in our system, whenever we are having those with the food, they are not properly utilizing, the system cannot absorb those things properly because of the high metabolic activity of these things. They are not soluble in water, so they are not present in our blood, and they are generally metabolized and excreted out very easily. So we find that a black seed extract, that is thymoquinone, it has a very high effect on cancer cell, killing cancer cells. So whenever they are targeting towards the cancer cells, the efficiency is just like any other chemotherapeutic agent that we have with us. But the main problem is that effect of thymoquinone is multivarious. A lot of papers are coming, lots of paper. But the main problem is that everyone is working in some system, everyone is doing this and that, and showing that in mice and in rat it is reducing. But I find something new, that it is a very much hydrophobic compound. So it is not soluble in water. So how could this molecule reach the specific area where I want to target those things? For that, it is also a light sensitive thing. For that, I have encapsulated the thing, a chemical. Generally, I am from a background of cell signaling molecule, but still for the last 3 years, I worked some on chemical synthesis, and I synthesized a new molecule like nanomolecular thymoquinone with pegylation, polyethylene glycol. And I find the synthesis procedure is just like that. I cannot explain this in a very short time, but I find that these molecules are very small, and through the FESEM and the transmission electron microscope shows that it's a very small molecule of less than 50 nanometer. 50 nanometer pegylated thymoquinone molecule. Pegylation and polyvinyl pyridine mediated encapsulation of this drug helps the molecule to release the drug very easily. The FTIR result and the UVV shows that inside the encapsulating molecule there are about 80% of the drug has been encapsulated, and the release effect is also very high. And we find the NMR spectra shows that each and every molecule of the synthesized polyethylene glycol has the drug inside. So these molecules can be used, these nanoparticle can be used against cancer, and we find that the size distribution, the size is very small, it is very much stable in different pH, and the in vitro, coming to the next type, that is the result that what I got in the in vitro. In in vitro, I find that it reduces the migration of cancer cells like MCF7, HBL100, and now I am working on triple negative breast cancer cells, but triple negative breast cancer cells show some different effect in different pathway. So there lies the problem, that here the scientists and the clinicians, so there requires a collaboration. There is a big gap between the scientists and the clinicians. Scientists are trying to find out all the signaling pathway. Clinicians are in the daily routine, they are doing the surgery, they are doing the chemotherapeutics, they are applying this drug and that drug. So the basic thing is that we're doing the experiment in the lab and they are doing the things directly on human beings. So whenever we are coming in contact with each other, we're finding that although there is a gap, still if the drugs that we are preparing in the lab, if they are being brought in the clinical handling by the clinicians to the human beings, then it would be very good, because the migration of cancer cell is being reduced by the thymoquinone itself at a very high dose of 2 to 4 microgram. And when I'm using that nanomaterial, that nanoparticle, it is doing at a 1 microgram per ml dose. And the effect is shown very well by the migration study through the transwell migration and other different types of studies. And next, why I targeted these things and how these molecules are affecting this nanomaterial affecting this migration, I find that the lamellipodia formation of the cells, this is a TEM picture, transmission electron microscope picture of live MCF7 and HBL cells, where I find that these molecules, whenever they are applied, the protrusions that the cell is extending whenever they are going through the matrix and they are invading the system by the integrin recycling pathway, this pathway has been very much hampered, and the legs or hands of the cells that are present, that are being very much reduced by the effect of this nanomaterial. And by finding out that this is a figure of actin cytoskeleton of the cell, and we find that actin molecules that are helping in the formation of the lamellipodia, these actin molecules are reduced and they are punctated from after the application of this nano drug. And to find out why this actin polymerization is taking place or not taking place in presence of this drug, I targeted the microRNAs. I found out that a lot of microRNAs are present behind these metastatic cascade. And to find out what are the microRNAs that are related with the application of this drug, and I have gone through different types of microRNAs, and I have shown only 6 data for here. They are very much relevant, MIR10B, 142, 116.1, and among the most important is microRNA 34A. So I find that in case of 2 types of cancer, which is triple negative breast cancer and triple positive breast cancer, in case of triple negative breast cancer, we find not less than 50% p53 mutations. And whenever I'm applying this drug in p53 wild type cells, they are also migratory, and I found microRNA 34 upregulation. And now I am working on microRNA 10B, and I don't want to properly disclose, but still I find a very good relationship between microRNA 10B and MIR34A, because these are contradictory. Because in case of p53 wild type cells, microRNA 34 is very much is down regulated, and 10B is very much up regulated. So I find the effect of microRNA 34, and this microRNA 34 has a background of p53. p53 upregulates this microRNA, and I find the application of the nano drug helps in upregulation of p53. And when I block this p53 with p53 alpha, I find no effect of this drug. So effect of polyethylene glycol coated thymoquinone nanoparticle, we find that there is a good effect on RAC1, because this RAC1 molecule is related with the migration of these cells, because the RAC1 helps in polymerization of this actin. And I find that MIR34A directly binds to the RAC1 promoter. This is not my exact finding, but these findings are being done by some other persons. I find that with the application of this drug, the RAC1 molecule is down-regulated, and that helps the polymerization of actin. And when I used microRNA 34A anti-sequence and I applied the drug, I find that the RAC1 protein is not upregulating. So this confirms that microRNA 34A is very much related with the downregulation of RAC1 and disruption of the actin cytoskeleton. So this may be a very good target for cancers which are migratory in nature. So overexpression of MIR34A I have done, and I have shown through the same and tame data that whenever I am giving an anti-sequence of microRNA 34A and applying the drug, it is not acting, but the drug itself is active very much whenever the cell is not treated with anything. So I have also validated this data in vivo and find out that the results are showing that a very good regression of the tumor at a dose of only 5 mg per kg body weight of the mice, and also it's showing that the spleen size has been controlled. And the most important thing that I am coming in the next slide is that it is giving a very good protection. That is important. Whenever I am using doxorubicin or cisplatin or these types of drugs or paclitaxel, they are hampering the system. But in case of this type of drug, it is giving a very good amount of protection to all the different types of blood parameters like LDH, ALT, AST, ALP, and different types of ROS generating molecules. So protection is much more important, as because if by going through any chemotherapeutic drug, if a person dies of its side effect, then it's of no use. So verification has been done in molecular in vivo, and we find the same result. In conclusion, I am showing that PEG 4000 TQ NP has effect on P53, which upregulates microRNA 34A, which helps in downregulation of RAC1, and depolymerization of the actin has been done. And this has a very good publication this year. It has been published in Biomaterials with a high impact of 9. And I hope that this thymoglobulin nanoparticle emerge as a potential effect of anti-cancer agent. And now I am working with this nanomaterial to be targeted to the specific site by binding with some specific antibody. So breast cancer, they express a lot of specific antigens. So if you can target that, and I'm working on those things with folic acid receptor and folic acid binding those things. So this is my group. They are working under my guidance. They are the 5 persons who are, and I would like to acknowledge different professors and all my lab mates and DST, DBT, Government of India for funding. Thanks.